Hello, quarantine world. This is group 2 coming at you with Angelman Syndrome yet again. We're going to be talking about the research and treatment options that are available today. This is brought to you by Kathy, Caitlin, Aaliyah, Van, and Allison. So a little humor to start off the presentation. Yes, Angelman Syndrome has been around for a while and there's no real cure. But don't be ignorant. People are working on it. And the future is bright. So here's a little introduction. So we've gone over most of this before, but Angelman syndrome is a complex neurological disorder, first described by Harry Angelman in 1965. There is no cure, sadly. So that's the main focus of this presentation, the work on that fact. The main cause of this is due to deletion of the maternal chromosome in that 15q region and causes a complete loss of function in that gene, the UBE3A gene, and that leads to dysfunctional cell ubiquitination activity. And in humans, there are 2 copies of the gene. The maternal copy is mutated Angelman syndrome, and the paternal copy is silenced, so that UBE3A gene is going to be expressed. And the parental copy is silenced whether you have Angelman syndrome or not, and so that's a real issue genetically with this. Research has been focused on improving the standard of living for patients primarily, but now that we understand and are understanding more of the cellular process behind Angelman syndrome, new research is promising for a cure.
So I'm going to talk about the current research that scientists are doing to cure AS. And so just to review what Van said, AS occurs because the UBE3A gene is mutated, and it's actually mutated on the maternal allele. So humans have 2 copies of the UBE3A gene. So in patients that don't have AS, the parental allele is actually silenced and the maternal allele is expressed. AS occurs when there is a mutation on the maternal allele. And so as you can see here under the AS syndrome, so the paternal allele is silenced, but the maternal allele is mutated. So therefore, there's not an expression of UBE3A gene. And the paternal allele is actually silenced by a long non-coding RNA. And so scientists are working with antisense oligonucleotides. So they're single-stranded deoxyribonucleotides, and they actually are complementary to the mRNA. So as you can see in the diagram, the ASO, it binds to the target mRNA, which in this case is the long non-coding RNA. And once it binds, it recruits an enzyme called RNase H. And what RNase H does is remove RNA primers. And if you can recall, RNA primers are actually like the first primers that bind to the DNA that signal DNA replication. So if the RNA primers are removed, then the enzymes will not recognize where to start DNA replication. So then this will prevent the formation of the non-coding RNA. And then a second technique that scientists are studying is using CRISPR and Cas9. So CRISPR and Cas9 is like a gene editing technique. And so what scientists are aiming to do is using this gene editing technique, they're going to go inside and edit the long non-coding RNA so that it doesn't silence the paternal allele. So as you see in the video, that represents the CRISPR, and it's going in, and that long rope around that allele or chromosome is actually the long non-coding RNA. So once you get rid of that long non-coding RNA, the paternal allele can be unsilenced, and it can start expressing the UBE3A gene. And then the last method that scientists are looking at is using topoisomerase inhibitors. So topoisomerase are enzymes that work during DNA replication. They help relieve supercoiled stress. So DNA is actually double-stranded. So when DNA replication occurs, you have to unwind that double-stranded, and when you unwind, you cause like a supercoiled stress, which is in the overwound region. And so what the topoisomerase does is that it breaks the DNA, it unwinds it so it can relieve the stress, and then it rejoins it. And so scientists are actually using topoisomerase inhibitors because this will inhibit topoisomerase from uncoiling the supercoiled stress, which will prevent DNA replication. and then furthermore prevent the formation of the long non-coding RNA that actually is responsible for silencing the paternal allele.
So I'm going to talk about an experiment that was posted in the Nature Research Journal that tested this idea of unsilencing paternal UBE3A genes through the administration of certain drugs. So in this experiment, they added yellow fluorescent proteins to the paternal UBE3A alleles in order to detect unsilenced alleles throughout the testing. So they started with finding the best topoisomerase 1 or top1 inhibitor, since there's a large variety of them that can all unsilence paternal alleles. So they tested the dose-response relationship between two different top1 drugs, and those drugs were irinotecan and topotecan. There we go. And they also tested inactive analog of CPT, and that was done because top1 is derived from CPT, and they wanted to see if it would also have an effect on the paternal allele. So in graph 1, you can see that as the dose of top1 drugs increased, UBE3A became more regulated, and as the dose of inactive analog CPT went up, it was not regulated. So the main thing that we can draw from this graph is that topotecan regulated UBE3 at a much faster rate than the other two. Graph 2 shows an additional test, and this was more concerned with duration of treatment and the levels of UBE3A in neurons. So topotecan increased the neuron level at a much higher rate for the same duration compared to irinotecan. And due to the information from both of these graphs, they determined that topotecan was at least 20 times more potent than the other drug. So then they tested this drug a little bit more. And if you look at the graph on the side, the first bar on the left shows the percent of UBE3A in a wild type. And the bar on the far right shows the percent of UBE3A when the maternal allele is deviant and exposed to topotecan. So you can kind of tell that those bars are almost the same level, meaning that topotecan almost completely restores UBE3A proteins to the wild type level. So for all of these reasons, they decided to use this drug for the rest of the experiment. So the next thing that they tested was different types of drug administration that would not only be effective, but also last a lot longer. So the first method that they looked at was intracerebrovesicular infusion, which is just the injection of substances directly into the cerebrospinal fluid in the cerebral ventricles. So this method proved not to work as well because only the infused hemisphere showed signs of unsilenced genes, and the topotecan concentration levels declined significantly only 5 hours after cessation of the drug due to rapid metabolism in the brain. And the degree to which the paternal allele is unsilenced is directly correlated with drug concentration. So when you have low levels of concentration, you're not going to have very high levels of unsilenced alleles. So they moved to the second method of administration, which was the intrathecal delivery, which is the introduction of pain medication that's usually more for spasticity directly into spinal fluid. So with the picture of the mouse, you can kind of see that this can also be used as an alternative way to deliver drugs into the brain. So this time, the topotecan was very well tolerated and increased the number of unsilenced paternal UBE3A alleles. And in the graph down there, you can kind of see that, yes, the concentration did drop slightly, but otherwise it stayed pretty elevated. And whenever it did drop, that was 12 weeks after cessation of the drug. So it lasted a lot longer than the other method did. So therefore, it was determined to be more effective. So why should someone with AS care about these results? Well, for one, it brings about the possibility of a long-lasting treatment, and therefore brings about the possibility of a chance of having a normal life. However, this idea of unsilencing the paternal allele is still relatively new, and so this course of action is still in the process of being studied, and the methods aren't perfect and there's very limited testing. However, it still brings possibility to treatment, and there are other options that AS patients can take when it comes to their symptoms.
So now I'm just going to go over a few prevention techniques and treatment options. So there is no way to prevent AS because AS occurs as a result of a genetic abnormality. And although most cases of Angelman syndrome are not inherited, particularly those caused by deletion in the maternal chromosome 15, which was talked about before, the risk of having another child with Angelman syndrome depends on the specific cause. So in order to take into account these risks, you will need to speak with a genetic professional to understand the risk in future pregnancies that are specific to your situation with Angelman syndrome. So there are a number of genetic tests that can be used to inform a couple about the possible outcomes of a current or future pregnancy with a child that has AS. Examples of two such tests are prenatal diagnosis and pre-implantation genetic diagnosis, which also goes by PGD. So prenatal diagnosis can be used to diagnose a condition in a developing fetus. If the underlying genetic cause of a disorder in a family is known, testing can be completed to analyze the genetic material of a fetus. This is typically completed around 10 to 13 weeks gestation via chorionic villus sampling, or around 15 plus weeks gestation via amniocentesis. Prenatal diagnosis may help patients prepare for the birth, or parents can also use this information to make decisions regarding whether or not to continue the present pregnancy with a child with AS at all. PGD is an alternative to prenatal diagnosis, and it is used following in vitro fertilization to diagnose a genetic disorder or condition in embryos. Only those embryos that do not carry the disease-causing mutation are implanted into the mother's womb. PGD allows testing to occur before a pregnancy even begins. In many cases, the disease-causing mutation must be identified first in an affected patient before PGD or prenatal diagnoses can be performed. So although there are many clinical trials of potential AS treatments in progress, there are currently no treatments for AS itself. The available treatments just focus on symptoms and helping the individuals find the best way to live with the disease. So although there's no specific therapy for Angelman syndrome at this time, the best treatment is to minimize seizures, anxiety, and gastrointestinal issues and maximize sleep. Seizures are treated with medications and dietary therapies, which will be talked to more about by Caitlin in the next slide, while sleep issues are treated with medications and sleep training. It is also important to test for and treat any problems with vision, hearing, and mobility. Intensive therapies such as physical, occupational, and speech therapies are critical to begin early and continue as long as necessary. Applied behavioral analysis and/or behavior therapy also are important for many individuals with AS. After early diagnosis, treating problems with vision, hearing, motor skills, and mobility with ongoing intensive therapies are also critical. A multidisciplinary team of healthcare professionals will likely work with AS patients to manage their condition, and depending on the signs and symptoms and the severity of those, treatment for Angelman syndromes may involve physical therapy to help with walking and movement problems such as ataxia that are most times lifelong issues for these patients, communication therapies, which may include sign therapy, picture communication, and gestures because most individuals with AS are never able to talk, and behavioral therapy to help overcome hyperactivity and a short attention span to aid in development.
Okay, so I'm going to talk about the mode of action for the anti-seizure medications. So first of all, GABA is an inhibitory neurotransmitter in the nervous system. So in a normal brain, GABA binds to a receptor which opens a chloride channel, causing the neuron it's acting on to not be able to depolarize, so it can't fire an action potential. And then over on the right, we see in an epileptic patient, GABA doesn't bind to receptors, so the inhibitory effect is gone, and the neuron can fire unregulated, and that's what causes seizures, is pretty much all or a group of neurons just randomly firing with no inhibition, and then you have a seizure. So specifically in AS patients, GABA isn't able to bind to the receptor, which causes the inhibition effect to be gone, which is why a bunch of the patients have seizures. So a doctor prescribing an anti-seizure medication for a patient with AS would need to specifically prescribe a medication that's a GABA agonist, so that the function of GABA can be rescued and the agonist can bind to the GABA receptor and cause the same inhibition effect as if it were GABA itself. So GABA also plays a role in memory formation and brain efficiency. So the first picture is kind of what our brains look like when we're newborns to one. All the different lights are different neurons firing at the same time for like to make a memory of what toy you like. So our brains at this stage are making as many connections as possible, as many synapses as possible, and then they get refined throughout our lives. So this would be probably about our teen years, where we still have a lot of connections that aren't really necessary to get from point A to point B, but our brains are starting to become more efficient. And then this last picture would be in our mid-20s, when our brains are at their peak and they're most efficient, where it takes maybe 3 neurons to get from point A to point B. So this happens over time by GABA being released by the neurons into synapses that aren't really needed to get to the final goal. And then that also allows excitatory neurotransmitters to be released in the synapses that are needed. So that, like a glutamate, it strengthens the synapses that we need to make that final like one pathway. And then GABA is able to be released and diminish the synapses that aren't needed. So giving an AS patient a GABA agonist medication would not only help with their seizures, but also help with their brain efficiency and hyperactivity and stuff like that.
So some future research in the works for AS includes further understanding the UBE3A gene and protein, since it's the main driver behind the disease and its pathology in the first place. And so there's hundreds, I think it's about 300 known uncatalogued UBE3A gene and protein variants. And so comprehensive mapping of the whole complex will pinpoint dysfunctional areas, understanding the most common mutations, will help researchers and healthcare professionals determine the most effective treatment plans, and understanding the different options of the way that the gene and the protein interact will be very helpful. Another field of future research are pinpointing the specific areas and pathways of the brain and nervous system. They're tied to AS. In our last presentation, Allison talked about some of the main parts of the brain that are affected, but they don't have specific, really detailed areas of where Angelman syndrome is affecting patients. And the top picture is for the first mode of research I was talking about. That's Dr. Lee from WashU in St. Louis. So he's working on the UBE3A research. And the bottom picture is Dr. Elgersma from the Erasmus Medical Center in Rotterdam. And he's working on the brain and nervous system mapping. And there are other areas of related neurological research, obviously. They're really ancillary to the unsolved origins of AS, mentioned the first two fields. And some of those other modes of research include, similar proteins, other Angelman syndrome symptoms, and other cellular processes that are similar but not specific. So the first two are the main modes of future research.
So now we're going to talk about a few plans of action. So there's some research on some possible cures that would look at unsilencing the paternal allele and understanding the different variants of the UBE3A gene.
So in AS, the maternal copy is the one that's mutated. It's important to understand that the paternal allele is not mutated, only silenced in the nervous system. So a really promising treatment plan would be to be able to unsilence the paternal allele in the nervous system so that it can rescue the mutated maternal copy and a correct protein of UBE3A can be expressed. And then it's also going to be really important that we understand the impacts of the UBE3A variants because there's so many different mutations of UBE3A that it can manifest as very different symptoms. And then understanding all of those and being able to identify all the different symptoms are going to allow us to be able to treat the general conditions of AS and then also be able to make specific treatment plans for different patients that have varying degrees of the disease.
So in summary, Angelman syndrome is uncured. It's a developmental neurological disorder and is caused by a loss function of the UBE3A protein.
Current research is trying to unsilence the paternal allele by using AOS, CRISPR, or Cas9, and topoisomerase inhibitors.
By using TOP1 inhibitors like topotecan, the paternal UBE3A gene can be unsilenced for possible AS treatment.
There is no standard treatment for Angelman syndrome, but instead doctors are focusing on managing symptoms to maintain the highest possible quality of life for AS patients.
And there's future research working to catalog UBE3A variants and pinpointing specific areas AS is expressed in the nervous system. So here are citations. Hope you learned something and talk to y'all soon.